All right, hello everyone. Welcome to Rachel Brooks's thesis defense. I'm Scott Hamilton, her advisor here at Moss Landing Marine Laboratories. Before we start, I just wanted to remind everyone about the etiquette for the Zoom defense today. So please stay muted during the defense. Don't try to share your screen or turn on your video. But then when the presentation is over with, everyone in the audience will have a chance to ask Rachel questions live. And so if you'd like to ask her a question, you can use under the reactions tab, which is either at the bottom or the top of your Zoom screen. There's a little smiley face. Click on that, and there's a button you can hit to raise your hand. Then we will call on you, and when you're called on, we'll ask you to unmute yourself. And at that point, please turn your video on so Rachel can see you. You can ask your questions, and then at the end, don't forget to go back and then hit that raise your hand button again to lower your hand. Once we're done with the question session, then we'll ask everyone to unmute themselves and turn on their videos and give Rachel a big congratulations. And then we'll kick everybody out, and the committee will stay and have a further discussion with Rachel. So do a bit of an introduction. So tell you a bit about Rachel's background. So Rachel just want to say from the start, was a really phenomenal student. And she'll be talking to you in a little bit about the research she did looking at the life history and demographic variability of this fish here, the Canary Rockfish, across the US West Coast. And it was just a tremendous amount of work. So a little bit about her background. Right, so Rachel, if you know her, she is, seems to be more aquatic in many ways than terrestrial. And from an early age, right, she's always around the pools and lakes and in the ocean, above and below the water as well, right? And you might see here that she at times does have a bit of an aversion, I think, to that terrestrial stuff, the dirt. Right, again, more evidence that she likes to spend a lot of time below the waves, right, spending time right in the ocean. Right, but there's a lot of evidence she was pretty gifted as an athlete, though, in both the watery and the terrestrial realm. So high school and collegiate water polo, and for any of you to know, this is a very grueling sport, and so I knew at that point that she would be very comfortable in the water and a really strong diver, right? But also in high school, track and field, doing discus, and then here shot put and pole vaulting. Pretty impressive stuff. So Rachel did her undergraduate degree at UC San Diego. So she worked as a grad student, Noah Ben Adderit, in my buddy's lab, Stuart Sandin, studying the movements and trophic ecology of this fish here, the yellowtail. And she continued to kick butt playing collegiate water polo while doing really well with her academic studies, right? And liking to frolic there in the kelp forest. And I met Rachel, I think it was sort of before a Western Society of Naturalists meeting here in Monterey. We were out at the Crown and Anchor. I was with Stuart, and he said, "Hey, I'd like to introduce you here to Rachel. She's going to be your future graduate student." And I was like, "Well, that's pretty presumptuous." And it was a little bit awkward now, but okay. And so we chatted for a while. And then when she submitted her application, I knew at that point she would be a great student. And so I think accepting Rachel was one of the best decisions I've made. Well, really, I guess I give Stuart credit. It was one of the best decisions he made for me. So Rachel took advantage of a lot of the opportunities that she had at Moss Landing. I think I had 4 classes with her. She did excellent in all of them. She was the TA for me and Mike for the marine ecology class. She took a number of the field courses that we offered that involve scuba diving, the subtitle ecology class, and the Baja class. I'll show a few images of that coming up. Right, she worked at the lab. She worked for Rick Starr and for me for the California Collaborative Fisheries Research Program. She also worked at the Marine Pollution Studies Lab. She was the co-chair of the Moss Landing Open House Committee one year, which is, again, a tremendous amount of work. She mentored a number of undergraduate students at CSUMB through the Undergraduate Research Opportunity Center program, and now currently through the REU program. She's had a number of awards and scholarships, the 2018 MLML Scholar Award, the CSU Coast Graduate Student Award, and the WAVE Award. Right, she also is great at communicating her science, and she gave presentations at 2 meetings, the Western Groundfish Conference, and as you can see here, us having fun at the Western Society of Naturalists meeting. So just a few highlights from her time here at Moss Landing. So she was in the Baja class trip where we went to Isla Natividad. She did her independent research project studying this fish here, the California sheephead, which I've studied for about 10 years. And so they're near and dear to my heart. And she got to do one of her favorite things, swimming around, collecting fish, cutting them open, looking at what was in their stomachs. And I should have known this because even at an early age, she was a pro at dissecting little sharks and fishes here, 6th grade, right? And this is basically, hey, she did this stuff for her thesis, right? But she also knew in the field, right, that you have to, especially in the desert, you've got to find a way to keep yourself well hydrated in the middle of dissections, right, or using kind of any implement you can find. Right, she also had an opportunity to participate in one of the 100 Islands Challenges expeditions. This is against Stewart's lab to French Polynesia. So she got to go to Moorea and Tahiti. I was on the leg after her. I was kind of bummed we didn't get to overlap, but here she got to go swim around in the tropics, again, collecting fish, doing surveys. And I think she realized that, oh, this is much nicer than the cold water and these thick wetsuits we wear here in Monterey.
Very much.
Yes. And then, you know, for a long time, she was one of the science crew for the California Collaborative Fisheries Research Program. And for the last year and a half, at least, she's been doing 2 jobs, not only finishing her thesis, but also working as the lead field scientist and then the statewide research coordinator for this program, 6 academic institutions across the whole state, coordinating hundreds of volunteer anglers and fishing boats and all the logistics and leading the trips and dealing with the data and the database and doing analysis and writing reports. I mean, she really has done it all. And we're really excited that she's going to be sticking around after she finishes her thesis, continuing to work for us for this program. Right, but all that hard work, again, occasionally catches up to her. So hard work, right? Work hard, play hard, occasionally crash hard. Right, so just a few more things about Rachel. Right, if you know her, she's very tenacious, right? She shows no fear, right? And as you can see here, right, she's not to be messed with. One example, the first time I went diving with Rachel, was still on the code for the subtitle ecology class. I think I was goofing around, you know, in between dives, and then I had to go to the bathroom. And when my wetsuit was down to my waist, Rachel pushed me off the boat into 50-degree water. And at that point, I knew I got to be careful around this one.
Oh God, I'll never.
Me either. But there's also some evidence that she, you know, she might want to try out for the TV show Fear Factor, right? Again, she has no fear. So one of her first fishing trips, Laurel and Bonnie convinced her she had to eat a lingcod heart if she really wanted to be a true fisheries biologist.
Her heart was beating still.
I have like that much memory on this.
All right, go and then tell me the story.
I mean, right, so we'll just finish off really quickly. All right, Rachel's not shy around the camera. She loves to pose with fish, as you can see here in all these examples. She'll pose with any other life form, right? She'll just pose, right, just for the fun of it, in lots of other amazing ways. All right, so we got about an hour to go. Rachel's going to tell you what she did for her thesis, and then afterwards, I encourage everyone to come. Let's celebrate Rachel and all her accomplishments. As you can tell, she really knows how to do that well. All right, and with that, Rachel, I'll hand it over to you.
Wow, well, thank you, Scott. I didn't know half those photos existed, so thanks for bringing those to light.
You have a lot of great friends and family.
Yeah. Okay, I'm going to turn off my video and start sharing my screen. All right, so you should be able to see my screen now. Yeah, so thanks, Scott, for that wonderful introduction. Very, very eye-opening. And thank you to the rest of you for joining me this morning as I present on a portion of my master's thesis entitled Geographic Variation in the Life History and Demography of Canary Rockfish along the US West Coast. Okay, so to prepare you for my talk, I'd like to give you a brief outline. I'll first start with an introduction of the major topics and concepts revolving around my study, and then I'll go into my research objectives. After that, I'll give you a rundown of the methods I use to complete these objectives, and then walk you through my results. We'll then move into the discussion, and I'll finish by providing you with a few of the main takeaways from my study. And then I'd like to finish this presentation by acknowledging those who made this project possible, and then I'll be ready for questions. And in case there's any confusion along the way, and to remind you of where we are in the presentation, I've provided these headings in the upper left-hand corner of my slides. All right, so let's dive into the introduction. So marine fishes with broad geographic ranges are exposed to gradients in environmental and oceanographic conditions, ecological factors, and anthropogenic stressors that influence their population dynamics. And for an increasing number of species, these factors have been shown to affect the life history and demography often resulting in altered population structure and latitudinal shifts in size and age structure, growth, maturity, and mortality. And so for my thesis, I wanted to examine geographic differences in the life history and demography of the Canary Rockfish along the US West Coast and provide updated spatially explicit life history and demographic data to aid in informing stock assessment decisions as well as resource managers. So before I jump into my specific study system, I wanted to define a couple key terms that are going to come up throughout the presentation and give you a bit of background on how the previously mentioned factors affect marine fish populations. So when I'm referencing demography, I'm referring to the population structure or frequency of age and size classes in a population, as well as mortality rates. And when I mention life history, I'm referring to traits such as timing of maturity or the size and age at which an individual becomes reproductively mature, as well as other parameters such as growth, longevity, or how long a fish lives, and condition or overall health. And there have been a number of studies of both fished and unfished species that have documented geographic variation in demography and life history. And often these patterns are associated with latitudinal gradients and environmental conditions. So generally, organisms inhabiting northern latitudes exhibit larger sizes, increased longevity, and mature later compared to organisms of the same species inhabiting lower latitudes. And this pattern in life history variability can often be explained by several ecological factors. It is generally attributed to temperature-mediated trade-offs between somatic growth and reproduction, as well as regional variability in food availability and metabolism. And the presence or absence through removal of large predators in an ecosystem can greatly influence the demographics of lower trophic level prey species. So where predators are more abundant, prey fishes often exhibit reduced longevity, smaller sizes at age, and mature earlier. In locations where the abundance of conspecifics are elevated and competition is intense, fish grow slower, attain smaller sizes, and mature earlier compared to locations with a lower abundance of conspecifics. And this was demonstrated by Cassell et al., who found strong density-dependent patterns at the population level of California sheephead across a substantial portion of their geographic range. So in locations with elevated densities of California sheephead, fish were smaller in size, had lower reproductive output, and changed sex earlier. And they attributed these density-dependent effects on life histories to resource competition and differences in prey quantity and quality among populations. And previous studies have shown too that fishing pressure can alter fish populations by removing typically the largest, oldest, and most fecund individuals, which often leads to a shift to smaller sizes, reduced ages, and earlier maturity. And conversely, fishing pressure has also been predicted to have the opposite effect in that fishery removals can alleviate competition, leading to greater food availability and resulting in faster growth. But overall, why is this important and why are we interested in how life history traits can vary across a species' geographic range? Well, it can have important implications on how we manage fish populations, especially ones that are broad ranging and occur across different environments. And for commercially and recreationally important fish stocks, stock assessments are used to inform resource managers when setting harvest levels. And these assessments essentially use models that are built upon estimates of stock biomass, exploitation rates, and quantifiable life history traits to monitor how productive a population is. So understanding the differences in life history parameters can help increase the accuracy of these stock assessments. And traditional stock assessments often don't consider spatial variation in life history traits due to the substantial complexity that it adds. However, ignoring these environmental variation may lead to misunderstanding the mechanisms regulating population dynamics and could potentially lead to a mismatch between harvest levels and stock productivity. And the US West Coast provides a really great system to study these environmental impacts on broadly ranging fishes, as this coastline is characterized by large gradients in sea surface temperature, with generally cooler waters up north and relatively warmer waters down south. The California current system runs parallel to this coastline and controls much of the productivity and upwelling in this region. And this range of coastal water encompasses a variety of habitat types, including both shallow and deep rocky reef habitats that are home to a wide variety of marine fauna, such as rockfishes. And rockfishes are a part of the US West Coast groundfish fishery, which formally includes over 90 different species of groundfishes. As many of you may know, rockfishes are long-lived, they're late to mature, making them highly susceptible to overfishing. And management decisions based on an incomplete understanding of rockfish life history led to the dramatic decline of 54 rockfish species in the late 1980s and early 1990s, and the resultant federal declaration of 7 rockfish species listed as overfished in the early 2000s. So to restore these historically low populations, harvest guidelines, quotas, trip and landing limits, as well as seasonal and spatial closures and gear restrictions were put in place by managers to constrain commercial and recreational opportunities. And stock assessments are conducted by the National Marine Fisheries Service, or NMFS, and they help to inform resource managers when setting catch limit decisions on groundfish species. And so these federal stock assessments integrate information from an array of fisheries, both dependent and independent sources, that are themselves strongly informed by the annual NMFS bottom trawl surveys, as most of the life history information is generated from biological samples collected during these surveys. And the trawl gear in this coastwide survey is excluded from high relief untrawlable habitats that these rockfishes are more often associated with. So it's possible that true population trends of many groundfish species are being obscured, and we need reliable sources of data from untrawlable habitats. So this brings me to my study species, the canary rockfish. And the canary rockfish occupy a large range across the Northeast Pacific, extending from Western Gulf of Alaska, south to northern Baja California, Mexico. But they're found most abundantly from British Columbia through Central California. And similar to many rockfish species, canary rockfish are known to undergo ontogenetic migrations, moving to deeper waters as they increase in size and age. So juvenile canary rockfish are commonly found in shallow subtidal and intertidal areas, while adults are commonly encountered along the continental shelf between depths of 80 to 200 meters. And canary rockfish occur in various habitat types, but are often found in and around areas of high relief. So because canary rockfish occupy a large range, this makes them and other groundfish species ideal candidates to study geographic differences in life history. And to provide you a bit of background on the canary rockfish fishery, so canary rockfish are both commercially and recreationally harvested along the US West Coast with other rockfish species. And this species experienced heavy fishing pressure, as I mentioned earlier, in the late 1980s and early 1990s, and were among the 7 rockfish species declared overfished in the early 2000s. So as a result, catches were substantially reduced, and retention was restricted for the recreational fleets across all 3 West Coast states. And the combination of stricter fishing regulations, as well as an updated assessment model and parameters, resulted in the canary rockfish population reaching above its management target, outlined here in green, and achieving a rebuilt status in 2016. So subsequently, new harvest specifications and regulations were implemented in 2017. And while there have been a few studies that have investigated canary rockfish life history in the past, few have accounted for canary rockfish across untrawlable high relief habitats. So that kind of leads me to the purpose of my thesis, which was to provide information regarding the life history and demography of the recently recovered canary rockfish stock within these undersurveyed areas. So that brings me to my research objectives. And my first research objective was to quantify and compare population structure and life history parameters of canary rockfish among latitudinal areas along the coast. And I hypothesized that demography and life history of canary rockfish would be variable along the coast. I predicted that as we transition from south to north, there will be an increase in sizes, ages, faster growth, and an increase in size and age at maturity, as similar trends have been reported for other groundfish species. I also hypothesized that there would be a gradient in body condition. Given that environmental conditions are variable along the coast, I predicted that canary rockfish condition would increase from south to north, as more productive waters in the northern extent of our survey area may provide increased availability of preferred prey, thus supporting better condition. Additionally, I hypothesized that natural mortality would differ among geographic regions, with lower natural mortality rates in the northern region, since older ages in the higher latitudes may imply lower natural mortality rates. And it also predicted that trends in total mortality will mirror that of natural mortality. Since leading up to the study, there were protections put in place to restrict catches of canary rockfish, decreasing the overall impact of fishing mortality. So my second objective was to investigate the associations of life history parameters with gradients in oceanographic conditions. And I hypothesized that the life history parameters of canary rockfish will be directly related to regional patterns in oceanographic conditions like sea surface temperature and productivity. And finally, my third research objective was to identify breaks within the coastwide canary rockfish stock based on similarities in life history traits. And I predicted that the breaks in canary rockfish populations will be latitudinally driven and occur at prominent biogeographic boundaries like Cape Blanco, Cape Mendocino, or Point Conception, as this has been seen in other studies conducted along the coastline. So now we'll move on to my methods, and I'll now go over how I accomplished all of this. So over the course of 2.5 years, we sampled canary rockfish out of 13 different ports, ranging from the US-Canadian border to just south of Point Conception. And the ports that we sampled out of are labeled on the map to the right, and you'll see that they're color-coded with those northern ports in cooler colors and southern ports in warmer colors. And I want you to keep note of this color scheme as it's going to come back up when I discuss my results. So we spent several days fishing out of each port with a goal of around 100 canary rockfish per port. And to catch our canary rockfish samples across high relief and trawlable areas, we used hook and line fishing gear aboard charter sport fishing vessels. And any bycatch that we encountered, which was most often other rockfish species, were released alive with the aid of descending devices. So in order to get to all these places, we needed a mode of transport. So we commandeered the white Moss Landing Marine Labs van, loaded up with an awesome crew of interns, a ton of fishing gear and science equipment, and hit the road. We traveled from California to Washington, and from there we worked our way back down the coast, sampling from port to port. And this entire project was a highly collaborative effort. So we had the expertise and advice of local captains, crew members, resource agencies, and academic institutions. We also had the help of over 250 amazing volunteer anglers from all up and down the coast that volunteered their time to come out on these trips to help us collect our canary rockfish. And while fishing on the boat, we obtained a suite of information such as number of fishers, GPS location, bottom depth, relief, and drift time. And the landed canary rockfish were tagged with a numbered T-bar tag, measured, and a fin clip was taken and preserved for future population genetic studies. And we did our best to try and collect a variety of canary rockfish, both small and large, female and male, and from both shallow and deep water habitats. So after our collections, we'd return to port for dissections. And oftentimes we had to get a little creative with setting up our mobile laboratory, which sometimes attracted the attention of the police for reasons I'm sure you can guess based on the top right picture. And I just want to take a brief moment to thank the incredible shop team, especially Chris, for designing and constructing the awesome portable dissection table pictured on the bottom right. It wasn't originally designed for me, but I got to use it in my thesis. And this table was just what we needed to conduct our dissections on the road. And it seriously has withstood the test of time, and it's been used for several different projects now. So thank you. And during our dissections, we obtained numerous biological metrics such as length, weight, sex, maturity status, and organ weights. We also collected muscle tissue samples and stomach contents. They weren't used directly in this study. And lastly, we collected and preserved gonads in 10% buffered formalin for later histological analysis, and extracted the fish's ear bones or otoliths for age determination. And a big component of my study consisted of determining the age of my canary rockfish samples. And there are several different hard structures and fishes that can be useful in determining their age. One structure that is commonly used in aging studies and was used in this study are otoliths, otherwise known as a fish's ear bone. And so pictured here is one of my otoliths in the center of the slide. And so to determine the age of my canary rockfish samples, I used a variation of the break and burn method. And this technique is what is predominantly used to determine the age of canary rockfish samples for stock assessments. So this process begins first by scoring the center of the otolith. And because of the robust size of the Canary Rockfish otoliths, I used a high-speed saw to split the otolith in half and then polish the cut edge. This was then followed by burning the exposed edge over an ethanol flame to increase the contrast of growth bands. I'd then place the burnt half in modeling clay, dab it with a bit of mineral oil before taking a look at it under the microscope. And I was really fortunate to spend a week learning the proper age rating process from the experts at the Northwest Fisheries Science Center Aging Lab in Newport, Oregon. And much like you would count the rings in a tree trunk to age a tree, you can count the annuli or translucent bands that form once a year during winter growth. So this is an image of a transverse section of a Canary Rockfish otolith, and light blue dots indicate annual bands used to age each fish. And this particular sample was aged to 18 years old. And as a method of quality control, I did a blind read of a subset of my otoliths to see if I showed any aging bias. And I also compared my age reads with the Aging Lab in Oregon to test for inter-lab bias and accuracy. And from this, I found that my age reads were within the accepted range of aging error. So moving on to my analyses, I've broken them up into 3 different groups based on my 3 research objectives, and my results will follow the same structure. So to compare demography and life history parameters of Canary Rockfish among latitudinal areas along the coast, I quantified a slew of different demographic and life history traits, including size and age structure, average oldest age, growth, size and age at 50% maturity, condition, and mortality. And I'll go over how these parameters were derived when I present on them in my results. I performed a principal components analysis to determine the spatial associations of coastwide Canary Rockfish life history traits and used those principal components in a linear regression analysis with chlorophyll A as a proxy for primary production and sea surface temperature to investigate the associations of life history parameters with gradients and oceanographic conditions. And the oceanographic factors that I used in this study were obtained from public NASA databases and selected from the closest catch location and averaged over a 12-year period for each port. Then to identify breaks in the Canary Rockfish stock based on similarities in life history traits among ports, I used an agglomerative hierarchical cluster analysis, and the average silhouette width was used as a means of statistical clustering validation to determine the optimal number of groups. I also ran a permutational multivariate analysis of variance, or PERMANOVA, to determine whether the variance in port clusters can be attributed to growth-based life history predictors. So with that, we'll get to my results. So this table provides a breakdown of the number of Canary Rockfish collected out of each port by sex and the depth ranges they were collected at. And so for the subsequent analyses, I pooled the sexes. I did look at sex-based differences in Canary Rockfish life history traits, but I won't be focusing on that for the purpose of today's talk. So if you want to learn more, I'd be more than happy to discuss this at the end of my talk, or you can check out my thesis for more information. And so from this table, I want to point out that the depths at which Canary Rockfish were collected were somewhat variable among ports. And this slide shows the relationship between depth on the x-axis and total length on the y-axis for the figure to the left and age on the y-axis for the figure to the right. And overall, we can see that both total length and age increased with increasing depth. So to account for this confounding factor of depth in my sampling, I used the residuals in my statistical analyses to evaluate the results of size and age structure among ports. So I compared the frequency distribution of sizes and age among ports using a non-parametric Kruskal-Wallis test. And for these figures, you'll see the frequency on the y-axis and the length on the x-axis for the figure to the left and age on the x-axis for the figure to the right. And so the red bars on each distribution mark the median size and age for that port. And ports are organized from south to north. So using the residual values from the previous linear regressions, I found that both length and age frequency distributions of Canary Rockfish populations varied considerably among ports. Following the red bars, we see a slight increase in median size and age from Central California to Southern Oregon, and an increase in median size and age from Southern Oregon through Washington. And we can see that this trend is much more pronounced for length and less so for age. So to statistically assess differences in longevity among ports, I calculated the average oldest age of the upper quartile of ages and compared among ports using an ANOVA. So this figure shows the average oldest ages for each port. And similar to the distribution plots, I have ports arranged from south to north along the y-axis and average oldest age on the x-axis. And from this figure, we see the average oldest age varied across ports. So letters above the bars denote significantly different groups. And we can see that Canary Rockfish from Washington and Northern Oregon ports tended to exhibit much older ages relative to Canary Rockfish from Southern Oregon and California ports. And I then used the von Bertalanffy growth function to compare lifetime growth trajectories among sampling ports. And essentially, the von Bertalanffy growth function is used to model the mean length at age of a population based on the size at age data. So the figure I'll be presenting shortly is going to look a lot like the one depicted here, minus the really beautiful Canary Rockfish pictures, where we have size on the y-axis and age on the x-axis. And like many organisms, most fishes exhibit rapid growth over the course of their early years. Eventually, their growth tapers off to the point where the fish virtually stops growing and reaches an asymptote, highlighted here in green. And this is represented by the von Bertalanffy growth equation listed above. And so the two parameters that are most often considered in stock assessments and were compared in this study were L infinity and K. And L infinity indicates the asymptotic average length, while K is an estimate that is used to predict how fast a fish will reach that asymptotic average length. And I also want to point out that both L infinity and K are inversely related, meaning for larger asymptotic average length, it will take longer for a fish to reach this size, thus a smaller K value. So this figure depicts the lifetime growth curves estimated from my size at age data for each port. And as a reminder, northern ports are colored in those cooler colors, while the southern ports are colored in the warmer colors. And what we can see from this figure is that the growth curves were variable among ports. So generally, we see that asymptotic average length, or L infinity, increases with increasing latitude. And we also see growth parameter K generally decreases with increasing latitude, meaning that Canary Rockfish from northern ports are reaching their asymptotic average length much slower than those from southern ports. And now the figure on the bottom right shows the 95% confidence intervals for the L infinity and K parameters, with L infinity on the y-axis and K on the x-axis. And so what I want you to get from this is that ports with overlapping lima beans indicate similar growth. So generally, we can see that growth parameters L infinity and K separate out into two distinct groups. The Washington, North Central Oregon, Eureka, Moss Landing, and Santa Barbara Canary Rockfish form one group, while Canary Rockfish from Southern Oregon and North Central California form the second group. And so then to estimate the predicted size and age at which 50% of the population is mature, I fit a logistic regression to the proportion of mature individuals for a given length. So my figures for maturity will look very similar to the example plot pictured here, with proportion mature on the y-axis and either total length or age on the x-axis. And so the horizontal dashed line in the center of the plot here will indicate the threshold for identifying when 50% of the population is mature. And the dotted vertical line will indicate the size where 50% of the population is mature for that particular curve. So here are the results for length at 50% maturity. And overall, we see kind of a latitudinal trend in which length at 50% maturity generally increases with increasing latitude, meaning Canary Rockfish are maturing at larger sizes as we move from south to north. So moving from those warmer colors to those cooler colors. Looking at the same figure, but with age on the x-axis this time, we see a relatively similar latitudinal trend where the age at which 50% of the population matures generally increases with increasing latitude. So meaning Canary Rockfish are maturing at older ages as we move from south to north. So to estimate the condition or overall health of my Canary Rockfish samples, I use two different metrics. I use the Fulton's condition factor, Fulton's K, and this is a metric used to compare how fat a fish is for its length. So for fish of the same length, higher values indicate fatter fish and better condition. So using these canary rockfish on the right as an example, we would consider the top fish to have a higher Fulton's K and be in better condition compared to the bottom fish. I also looked at the Hepatosomatic Index, otherwise known as HSI. And this is a metric that looks at the liver to body weight ratio and can be used to infer the lipid storage or energy reserve status of a fish. So higher values of HSI indicate greater energy reserves in the liver. And using the canary rockfish pictured on the right, again, as an example, we would consider the bottom fish to have a higher HSI and be in better condition compared to the top fish. And so both these metrics were derived for each sampling port and compared using an ANOVA. So now we'll review the results from my condition analyses, starting first with the morphometric index Fulton's K. So to orient you to this figure, we have Fulton's K on the y-axis and ports arranged from south to north along the x-axis. And generally, we see that Fulton's K values were variable among ports. Once again, the letters above the points denote significantly different groups. And we found that canary rockfish from southern ports exhibited slightly higher Fulton's K values, meaning they were heavier on average compared to the canary rockfish from other sampling ports, with the exception of Newport and Eureka. Now swapping out Fulton's K with the Hepatosomatic Index, or HSI, we generally see the opposite pattern. So with larger standardized liver weights observed across canary rockfish collected from Westport, Garibaldi, and Newport compared to all other sampling ports. So lastly, I compared mortality rates among ports, and specifically I looked at natural mortality and total mortality, which is essentially the sum of natural and fishing mortality. And natural mortality was measured using an empirical equation that's based off on maximum age, so similar to a technique used in the recent stock assessment. And total mortality rates were measured or were assessed through age-based catch curve analysis using log linear regression of the age frequency data from each port and excluding fishes younger than the modal age. And then ANCOVA was used to statistically assess differences in slopes among ports for my total mortality rates. And to better visualize these differences, I plotted the total mortality rates as point estimates for each port. So now we'll review the results from my mortality estimates. So similar to the condition plots, I've arranged the ports from south to north along the x-axis. And we have natural mortality on the y-axis for the top figure and total mortality on the y-axis for the bottom figure. So looking at the natural mortality rates first, we see that there is some variability among ports. And generally, we see natural mortality rates are slightly lower across the northern ports. However, since natural mortality is calculated using that maximum age, it tends to be a bit more sensitive to outliers. Now looking at total mortality rates, so we can see that total mortality rates differed significantly among ports and shows a relatively similar trend as natural mortality rates, where total mortality decreases slightly from south to north, with mid-range ports tending to be a bit more variable. So now that we've quantified and characterized our site-specific demography and life history characteristics, we can now look at their spatial associations within or with oceanographic parameters. So to examine the spatial associations of coast-wide canary rockfish life history traits, I ran a principal components analysis, or PCA. And a PCA is a multivariate analysis where you can essentially take a set of variables, in this case, canary rockfish life history traits, and reduces them into a smaller set of principal components that are still able to illustrate the important patterns in the data. So I selected the following life history traits based on parameters that are important to fishery stock assessments. And when interpreting a PCA, it's important to note the magnitude and direction of the vectors, as this indicates how important a particular variable is to the component. So the direction of the vector in relation to one another tell us how the life history traits are associated with one another. So for example, if you look at L infinity and K, you'll see that they're opposite from each other, which shows that they're inversely related. And finally, each point within this figure represents a sampling port. So with the northern ports in cooler colors and southern ports in warmer colors. And overall, principal component 1 and principal component 2 together accounted for up to 78% of the variation seen in life history data generated from this study. So principal component 1 accounted for approximately 57% of the variation and was driven primarily by factors related to age, growth, and maturity. So positive values on PC1 are correlated with average oldest age, L infinity, and maturity, while the negative values are correlated with the von Berlanti growth parameter K and natural mortality M. And principal component 2, or PC2, accounted for approximately 22% of the variation and was primarily driven by the condition index Fulton's K on the positive scale. So to examine the relative influence of long-term oceanographic conditions on life history traits, I saved the principal components to use in a linear regression analysis with mean chlorophyll A as a proxy for productivity and mean sea surface temperature, or SST. And again, each point on these figures represents a sampling port. And so for both figures, we now have the principal component 1 values on the y-axis and the mean sea surface temperature on the x-axis for the figure on the left and mean chlorophyll A on the x-axis for the figure to the right. So zooming in on the figure to the left, we can see there's a negative relationship between PC1 and mean sea surface temperature, suggesting that canary rockfish from colder locations grow larger, live longer, mature later, and have lower natural mortality rates compared to fish from warmer locations. Now zooming in on the figure to the right, we can see there's a positive relationship between PC1 and mean chlorophyll A, suggesting that canary rockfish from more productive locations grow larger, live longer, mature later, and have lower natural mortality rates compared to fish from less productive locations. So now swapping out the principal component 1 with principal component 2 values, we'll now focus in on the figure to the left with mean sea surface temperature on the x-axis. And generally we see a slight positive trend between PC2 and mean sea surface temperature, suggesting canary rockfish from warmer locations were in better condition or fatter compared to canary rockfish from colder locations. However, we found this relationship to be non-significant. Now focusing in on the figure to the right, we see that there really is no relationship between PC2 and mean chlorophyll A. So to identify subpopulations based on similarities in life history traits of canary rockfish among ports, I ran an agglomerative hierarchical cluster analysis, and Euclidean distances were calculated among ports using the same normalized life history parameters as in the PCA. So as the name itself suggests, this analysis essentially groups a set of objects in such a way that objects in the same group or cluster are more similar to each other than those in the other groups or clusters. And we can visualize the results from this analysis using a dendrogram. So the figure on the left depicts the dendrogram from the hierarchical cluster analysis comparing port-specific life history traits. So 2 clusters were identified as being the optimal number of clusters using the average silhouette width as a means of statistical clustering. And from the dendrogram, we can see that canary rockfish life history parameters derived for each port separate out into a northern and southern cluster, with a break in life history parameters occurring generally around Central Oregon between Coos Bay and Newport. So moving on to the discussion, I'll now tie it all together for you and move on to some key points or some key takeaways from this study. So we found canary rockfish demography and life history traits varied along the coast, right? And as previously hypothesized, we generally saw sizes, ages, growth, and size and age at maturity increase from south to north. We also found total natural mortality to vary along the coast, with both mortality estimates slightly decreasing from south to north. And contrary to what was previously hypothesized, we found canary rockfish condition to be variable along the coast and showed conflicting patterns depending on upon the method used. And we also found that Canary rockfish life history parameters are closely linked with regional patterns and oceanographic conditions. And we were able to identify a division near Central Oregon in the coastwide Canary rockfish stock based on similarities in these life history parameters. So what might be driving the patterns we see in Canary rockfish life history along the coast? So as demonstrated by the PCA and linear regression analysis, we saw that Canary rockfish life history parameters were closely linked with regional patterns in sea surface temperature and productivity. And we found Canary rockfish associated with colder, more productive locations grew larger, lived longer, and matured at larger sizes and older ages and had lower natural mortality rates compared to Canary rockfish associated with warmer, less productive locations. And so counter gradient variation in growth and size selective winter mortality may provide a potential underlying mechanism to explain the patterns we see in Canary rockfish growth, size, and reproduction. Counter gradient variation is a form of local adaptation that may evolve to counteract detrimental environmental influences on an organism's fitness. So the colder temperatures and shorter growing season associated with the higher latitudes along the Northeast Pacific may lead to the genetic selection of faster growth and larger sizes in northern populations through time. Juvenile Canary rockfish that maximize their growth and attain larger sizes during the shorter growing seasons in the north may be less susceptible to mortality from energetic deficiencies or predation as winter proceeds. Additionally, increased productivity in the north could support increased resource availability and preferred prey resources, which may also support increased growth and larger sizes observed in the northern populations. So preliminary examination of Canary rockfish diet contents revealed that teleost dominated the diet of fishes caught out of Washington ports, while euphausids and shrimps dominated the diet for Canary rockfish caught from Oregon and California ports. But it's also important to note that the stomach contents only provide a snapshot of what these fish were eating during that time and may not be representative of their entire diet. Depth could have also played a role in driving some of the variability in life history traits we saw. So as I mentioned in the introduction, adult Canary rockfish generally move into deeper waters as they increase in size and age. And we saw this pattern reflected in our sampling, with larger Canary rockfish caught in deeper waters. And although we found significant differences in population structure of Canary rockfish along the coast after accounting for the confounding factor of depth in our population structure analyses, it's still plausible that the maximum depth surveyed could have potentially skewed the longevity or my longevity estimates or natural mortality rates, as both these parameters are highly dependent upon oldest ages sampled. And in addition to environmental factors, historical fishery removals could potentially provide a source of variability in life history trends. So fishing pressure is often selective, as I mentioned at the beginning of this talk, in that it typically removes the larger, older reproductive individuals. And over time, this may lead to a downward shift in sizes and earlier maturation. So greater fishing pressure in the south could skew the size distribution of the population in that region to smaller sizes, thus reducing the southern estimate of asymptotic size relative to the north. However, historical catch reconstruction for Canary rockfish reported in the most recent stock assessment doesn't really corroborate this trend. So overall, they reported greater catch from Oregon, highlighted in red, followed by Washington, highlighted in green, and California, highlighted in red there, for all fleets combined. But it's also important to note that much of the sampling of Canary rockfish occurred when there were protections put in place to reduce the overall fishing pressure. So this suggests that environmental factors may have played a bigger role in driving the latitudinal patterns we see in Canary rockfish life history parameters along the coast. And zooming out onto the bigger picture, how do the results from this study compare to previous assessments on Canary rockfish, and what might be the broader management implications? So the results from our cluster analysis suggest there may be a division in Canary rockfish life history parameters just north of Cape Blanco. And this division doesn't necessarily align with how Canary rockfish have been assessed in the past. So the most recent assessment employed a spatially stratified model to account for spatial variation and exploitation history among states to produce a coastwide estimate of spawning stock biomass. But often state boundaries are used to designate areas in stock assessments, as they tend to have their own unique exploitation history. However, it's also important to consider the biological differences in the population when modeling stock productivity. And the evidence presented here based on the latitudinal variation in biological parameters may indirectly imply that Canary rockfish may exhibit some sort of stock structure along the US West Coast. And this should be further explored through DNA studies and potentially accounted for in the future assessment. Additionally, it's important to note that both hook and line and trawl fishing methods have their own limitations. So the hook and line fishing method allowed for collections across these high relief and trawlable habitats where Canary rockfish tend to frequent more, while the trawl fishing method accounted for samples across these low relief trawlable habitats. Conversely, the hook and line fishing method tended to be much shallower compared to the trawl fishing method. So there could be larger, older fish in deeper waters that weren't accounted for in this hook and line sampling, but are captured maybe in the trawl sampling. But regardless, both fishing methods are only really providing a snapshot of the population or a portion of the population, and we're missing a complete view of the entire Canary rockfish population structure. So potentially employing both these sampling methods might be the key in getting more robust estimates of population parameters. So with that, I want to summarize some key points from this study. So overall, the results from this project contribute new information regarding groundfish life history and provides updated spatially explicit estimates of life history parameters for the recently recovered Canary rockfish stock across these undersurveyed, untrawlable habitats. We found that geographic differences in demography and life history parameters can be very prominent in these broad-ranging species, especially for Canary rockfish. And often it can be difficult to disentangle the mechanisms behind what may be driving these differences. However, in the case of the Canary rockfish, regional effects such as gradients in temperature and productivity could drive the differences in life history traits observed here. The life history parameters generated in the study suggests a division in the coastwide Canary rockfish stock around Cape Blanco, and I suggest that future research explore stock structure through DNA studies. And as I mentioned before, we took fin clip samples from all the Canary rockfish surveyed in this study, so it might be a great master's thesis for a potential moss landing student. Additionally, both sampling methods only account for a portion of the population. So I think that the next steps in this research should compare life history characteristics between the samples collected in the trawlable and untrawlable habitats so that we can better understand how life history parameters may differ between these two sampling methods. And finally, short-term research projects like this one are informative and necessary to provide updated life history information for use in stock assessments. However, they only provide a snapshot of the population at one point in time. So to be able to track how populations respond to environmental and anthropogenic stressors over time, continuous long-term monitoring is needed across both trawlable and untrawlable habitats. So with that, I would like to go into my acknowledgements. As Scott mentioned, this was a really, really big project and it required the help of so many different people. So before we go into questions, I just want to take a brief moment and thank all the incredible people that helped to make this project possible, as well as those who supported me throughout graduate school. So first off, I'd like to thank the project PIs for being the scientific foundation behind this project. And I'd also like to thank Betty Kamikawa and Patrick McDonald for welcoming me into the CAPS lab and taking the time to train me on the proper age reading process. And I probably should have mentioned this in the introduction, but Canary rockfish have been validated to reach almost 80 years old or 84 years. So they're an old living fish and there were some very old samples to count. So I just want to extend a huge thank you to them. I'd also like to thank CCFRP and the ODFW Marine Reserves team for assisting with the sampling effort. And I'd like to thank the incredible captains and crew that had that collaborated with us. This project would not have been possible without their expertise and guidance. I'd like to extend a huge thank you to the Moss Landing community for their support throughout the years. You have all helped make this research facility run like a well-oiled machine. And I'm grateful for each and every one of you. Michelle, I really enjoyed working with you for over the past couple of years. You always have such a warm demeanor, and I truly appreciated your words of encouragement in our conversations. James, thank you for always checking in on me during the weekends or late nights at the lab. You were always there to just make sure I was okay. So I appreciate that. Chris, I just want to echo again some of my previous lab mates and say thank you for fabricating that portable dissection table. As I mentioned, it's withstood the test of time. It's seen a ridiculous number of fish carcasses, and I imagine it'll see that into the future. So thank you. I'd like to thank IT for making this transition to working virtually practically seamless. Kara, thank you for fielding my many grad school questions and always having an open door. So thank you guys. And the backbone of this project was undoubtedly the vessel captains, crew, and definitely the volunteer anglers. This project would not have been possible without their expertise and guidance along the way. I'm so incredibly grateful to have had their help. This project was truly, truly, truly a collaborative effort. I learned so much from each and every one of them, and I made so many friends along the way. Like, one of my fondest memories is going out of Brookings and Phyllis making like a full-blown breakfast with scrambled eggs and bacon and sausage on the fillet table as we're like steaming out to our fishing site. There's just so many incredible memories like that that I will never forget. Scott, I want to thank my primary advisor, Scott, for accepting me into this lab. I've learned so much from you, Scott, throughout my time here at Moss Landing. And I've been here for a while. So I started in Scott's lab back in 2016. And since then, I feel like I've grown so much as a scientist, but also as a person under his mentorship. He's always there to ask the hard questions and encourage me to think critically about my results. And he's super fun and awesome to learn from in the field. So not only did he come out on many of my thesis sampling trips and helped descend the many rockfish that were bycaught, but I also had the pleasure of working in the field with him for countless Moss Landing courses. I think this is a picture of him down in Baja diving for that sheephead project. And I'm going to get you back on this one. So I think my lab mates will all agree with me when I say that he has an incredible work ethic, and he truly embodies that kind of work hard, play hard mentality. So clearly you can see that I've learned everything I can from this gentleman right here. I'd also like to thank my committee members, Rick, James, and Gita, for providing valuable insight and supporting me throughout the graduate school process. Rick, your knowledge and expertise regarding groundfish and fisheries have been invaluable to my thesis. And thank you for teaching me how to think like a fisheries scientist and always challenging me to think critically about my methods. James, thank you for always reminding me to think big picture and placing my thesis in the broader context. And then finally, Gita, thank you for being a voice of reason in this group. I learned a lot from you in stats class. I just always appreciated your support and encouragement. It is always fun to run into you in the hall and just chat. So in addition to the many anglers that helped on this project, I also had a ton of help from my field research team, and this really would not have been possible without them. So Travis, Isidro, Erica, Heiju, Joseph, and Connor, these were my research interns. They helped me for both years of this study, and I'm so incredibly grateful to have had their help throughout this project. Not only did they work their butts off running, measuring, and descending fishes during our long fishing days, but we spent even longer days dissecting all those Canary Rockfish samples. And they were a huge asset in doing that. Yeah, probably more importantly though, they put up with my really poor choice in music on our travel days. And I'm not one to shy away from singing. And so they had to hear me sing off tune like every time we moved from port to port. So thanks for putting up with me, guys. And then finally, or almost finally, I'll get through this quick. I want to thank my Moss Landing peers for their support and their camaraderie throughout the years. Many of you offered a helping hand with my sampling trips. I would especially like to thank my fellow ichthyology and fisheries and conservation biology lab mates, especially at fisheries, just for like adopting me into your lab. I kind of like just forced my way through, so thank you. Both past and present too. Just for your advice, your mentorship, and your friendship throughout my time here at Moss Landing. I've learned just so much from each and every one of you, and I'm just incredibly grateful. And these are just a few of the many pictures we have to remember our time here. Okay, and then finally, I really would like to thank my family. So David, my dad Paul, my mom Rhonda, Coda, our little black lab, and Larry. for just their constant support and encouragement throughout my time at Moss Landing. Thank you for constantly asking me when I'm going to finish grad school. I really appreciated that over the last 5 years. So thank you. And just, being my support system and cheering me on and throughout everything. So yeah, thank you. Okay, next. And with that, I'll take questions.
All right. Great job, Rachel. That was a fantastic seminar. Thanks so much. And yes, you got me back indeed with that onesie shot.
I was more for the Nereocystis bong, but either way.
Yeah. All right, time for questions. Who has a question for Rachel? So you can go to the reactions tab at the base of your Zoom screen and click to raise your hand and we'll call on you. Maybe you've answered everyone's questions here. I'll ask you a question while we try to encourage other people to do the same. So, right, you weren't able to collect the fish all at the same time, right? I mean, you went over this huge geographic scale. And so some fish were caught in different times of the year. So what effects do you think that might have had on any of the life history traits and things that you measured?
Yeah, that's a really good question. Yeah, so there was some seasonal variability, which most likely influenced my condition indices. I'm not sure how reliable those trends are just based off the different seasons that we caught them in along the coast. So most likely influenced the condition indices.
And when do you think, I guess, they would have been in better condition? Like, if you caught them when, in the winter or the summer or the spring or
probably the summertime, which was a bulk of our sampling. But yeah, there were some opportunistic, or I guess some opportunistic sampling, in that there were a couple ports that we just kept getting weathered out of, and so we had to keep pushing off sampling during that time. But yeah, mainly summer, fall.
All right, thanks. So I saw Lauren put her hand up first. Lauren, you can unmute yourself and turn on your video. Ask Rachel a question.
Hey, Rachel, that was a fantastic defense. You mentioned super briefly that you had looked at the sex-specific data, but you ran out of time to cover it. I was wondering if there were any cool findings from that area of your thesis.
Yeah, thanks for asking that. Yeah, a big portion, or a big point of doing this research was in the trawl surveys. There is there's just a lack of older females, which have led the stock assessments to kind of have to assume a higher natural mortality rate for females. And so the thought was that maybe these older, larger females are hiding out in these higher relief rocky reef habitats. However, we didn't really find that in the results or in my data, which was surprising.
You could also mention, Rachel, what other differences did you see consistent differences did you see between the males and females in their life history traits along the coast?
Yeah, I mean, females grew faster, grew larger than males. Similar, I did find that females and males matured at similar sizes. The hope was to do to look at my ovary samples and do some histological work with that. However, just with COVID and things, I didn't really have the time to incorporate into that into my study to fine tune my maturity identification. So it was all macroscopically done.
All right, so some of your committee members have their hands up. I think I saw Rick's hand up first, so I'll call on him.
Hi, Rachel. Great job.
Great shirt.
Yeah.
So patriotic today, Rick.
This is for you. Yeah.
Getting ready for the 4th.
Yep. So you mentioned that you were surveying the untrawlable habitats, the higher relief rocky areas, and I know that NIMS surveys the untrawlable habitats from every year, basically. So I was wondering if you think that your conclusions would change much if you combined the two data sets if you combine some of the information from trawl surveys with your surveys?
That's a great question. I mean, it would just bolster my sample size, which would be really cool. I know there was a recent study published that used this trawl data to look at kind of latitudinal variability along the coast. That particular study used samples like across the past like 13 or 12 years of data that they have for those trawl samples. And they found some latitudinally driven differences with larger sizes up north versus down south. But it would be, so I don't know, I guess it relatively would be similar. We kind of had different methods of determining differences along the coast. So I don't know, I think it would be interesting to do a comparison or to combine both data sets.
Okay, thank you. Great job.
All right, James has his hand up. You can unmute yourself, James.
Yep, Rachel, really nice job. Well, really well organized. A lot of work, obviously. So you kind of alluded to this. Scott asked a little bit of this, and Rick talked about it. But huge geographic range with a lot of temporal variance, and you talked about in your discussion that there are some challenges of assessing communities across that kind of scope and that kind of time. Yeah. Just out of curiosity, to what extent do you think the results you have from your geographic range would be a proxy for any other species across that similar range? Like other animals that were caught coincident when you were doing your study? Do you have any thoughts about the extent, assuming that you couldn't do what you just did for canaries for everybody else, to what extent does this inform other species?
That was a great question. Yeah, we did keep track. So during our sampling, we did keep track of bycatch. So we have all that information of what species we encountered when we were catching these canary rockfish. And we also kept track of, we measured every fish that we encountered. So we kind of have some idea of length distributions along the coast of different species. We have a lot of that information for yellowtail. And they showed a very similar trend. I guess I don't know if I'm answering your question.
No, that's an example. Yeah, okay, great, thank you.
All right, any other questions out there for Rachel? I'll ask you one more while we give people a chance to see if there's any other burning questions. So, I mean, you mentioned potentially some of these life history differences may be driven by sort of genetic differences, right? And some adaptation, I guess, of the rockfish populations to different, thermal or productivity environments along the coast. And so what do we know from past studies about the population genetics of canary rockfish? Do they show different genetic breakpoints? Is there structure along the coast that might sort of help bolster that hypothesis?
Yeah, so there's a couple studies that have been done. Honestly, in my opinion, it's just still inconclusive because there was one study that showed some sort of or some genetic structure along the coast. But there have also been other studies that haven't really showed any genetic structure. So I don't know. I think the jury's still out and that we could potentially still answer that question.
Yeah, so it depends on the genetic markers, I guess, that you use.
But yeah, they all use very different methods, so it's kind of hard to tell.
All right, Rick put his hand back up, so we'll let him ask one more question and then we'll wrap it up.
So this was a really expensive study, covered a large geographic area and required a lot of people to be successful. Of course, a lot of your hard work as well. And with that in mind, when would you think that you would want to do this project again? Or when should we do this project again? You probably don't ever want to do it again. And in other words, how often do you think that these characteristics might change, especially with respect to changing climate?
That is a great question. So like what, yeah, that's a good question. I don't know if I can give you an exact timeline of like when to, or to keep like frequency of sampling. I know the trawl survey provides a really good data set in that it's a long-term project. So they sample every year. Like you said, this is very expensive. I don't know. I don't know if I can answer this question. I'm gonna have to think about it a little bit. I don't think it needs to be nearly as frequently as the trawl surveys are, would be my.
Okay, let me ask it a different way.
Yes.
How rapidly do you expect some of these characteristics to change with the changing climate?
Oh, not rapid. I guess like every 10 years, maybe. It's not like a year to year. There's not going to be year to year variability in my opinion. 10 years, maybe, if you want a number. You're muted, Rick.
Still muted. I have to ask him to unmute. There you go.
Yeah, no, I was saying I don't have a number in mind. I was just wanted to tap into your intelligence and expertise and see what you thought.
My best guess would be 10 years, but I don't know. Maybe a little, I don't know, maybe shorter.
Okay, thanks.
Okay, and then one final question in the chat. This comes from Shelby Ziegler. She says, what were the oldest canaries caught and what port were they from?
Oh, that's a good question. Yeah. So the oldest canary that I aged was 51 years old, and I believe that was out of Nia Bay, so our northernmost port. And then I think the next to that was like 40. Well, after that, Westport with 41, and then so on.
All right, great job, Rachel. So we can ask everyone now to unmute themselves. You can turn on your video. You can tell Rachel congratulations, and then the committee will meet with her separately.
Woo! Good job, Rachel.
Woo! Yay!
Congratulations.
Congrats, Rachel. You killed it.